Okay, got some technical problems going on.
Sorry.
That's okay. Am I unmuted? Yep. Okay. So today we have the pleasure of being joined by Dr. Liz Mallot. Dr. Mallot is a new assistant professor in the Department of Biology at Washington University here in St. Louis. She completed her PhD in biological anthropology at the University of Illinois Champaign-Urbana. So welcome back to the Midwest in 2016, followed by a postdoc training at Northwestern University, Dartmouth College, and Vanderbilt. Liz's research focuses on the causes and consequences of variation in the gut microbiome. Much of her research examines how diet, environmental change, and host physiology impacts wild non-human primate gut microbiome. But her recent work includes identifying factors shaping microbiome variation in adults and children in the United States. So thank you so much for joining us. I'm really excited to hear your talk.
Thanks. Okay, so I think many of you heard me talk about human microbiome work last year. I think so.
Okay. Yeah.
Cool. Can everyone hear me okay? Okay, great. So yeah, a lot of you heard me talk about my human microbiome work last year, but today I'm going to be talking about the non-human primate work I do. And I missed out on this experience of giving a seminar in this room as part of my job talk, so now I get to have it. So I'm going to be talking about some ongoing work in our lab. These are some messier stories that have resulted in more questions than answers. But I think they've prompted me to go in a really interesting direction that's kind of fun and exciting. So stick with me if it seems like I'm meandering around a little bit at times. I do have a point that I'm going to get back to at the end. So my lab studies host-associated microbial communities. These are the bacteria, archaea, virus, protists, fungi, and other microscopic eukaryotes that live in the gastrointestinal tract. These microbes are constantly interacting with host physiological processes, and many of their interactions with the host are beneficial, or at least not harmful. And we're really interested, because of all these beneficial interactions, really interested in understanding sort of what factors shape microbiome community assembly in the gut. Partly so that we can better understand how the gut microbiome contributes to host health and fitness. So there are multiple factors that play a role. A lot of environmental factors, diet, social factors, as well as some factors that are specific to the host, physiology, genetics, as well as life history stage. And the relative importance of these different factors is really highly variable across species. But understanding all of the interactions between these social and environmental factors is really important for understanding the evolutionary processes and ecological processes that shape microbial community structure. And it's really important for understanding the evolution of host microbe interactions. And I think about that in the context of non-human primates. Most of my work has been looking at how diet structures the gut microbial community, both within and across species. And I'm in part interested in diet because I have a background in foraging ecology. I've spent a lot of time thinking about how food availability and distribution in environments structures monkeys' decisions about where and what to eat, and subsequently how that influences that monkey's energetic status. But the foods an individual eats determine what nutrients are available to the gut microbes in their gut. And so then the composition of the gut microbiome is highly dependent on diet, but also which gut microbes are in the gut also impacts which nutrients are available to the host. So it's this sort of feedback loop where the host is influencing the gut microbial community and the gut microbial community is influencing host energetic status. So there are a lot of different dietary patterns we see across animals, and these dietary patterns relate broadly to differences in physiology, body size, body composition, morphology, habitat, all of these sorts of things that all also impact the gut microbial community. How much space is in the gut and what the pH and temperature of the gut is determines which bacteria would like to live there and how much space for them there is. So one of sort of the big open questions in studies of host-associated microbiome communities is whether diet or phylogeny has a bigger impact on the structure of the gut microbiome across species. And this answer really kind of depends on what data set you use and whether you're looking at captive or wild animals and what scale you're looking at. This is not a new question. We've been asking this question since before I started graduate school. Most studies find that the answer is both diet and phylogeny contribute, but the diet matters more when you're looking at closely related species, and that phylogeny matters at larger scales. A primate gut microbiome is always going to look like the gut microbiome of other primates, even when they are consuming a diet that is different from that of other primates. If they're consuming a lot of meat, they still look like a primate. Even when we look within primates, we see that phylogeny broadly structures the gut microbiome. We see that primates fit this pattern of phylosymbiosis, where the gut microbiome is more similar within closely related species. So all of these adelines here in green all have closely related gut microbiomes. The hosts are all in the same family. All of the lemurs here in purple have closely related gut microbiomes. And we see closely related gut microbiomes in Cervipithecine monkeys, teal. And we see this pattern repeat across primates. But then we have these red and orange folks over here. These are humans, and humans disrupt this pattern. Humans, you would think, would be most closely related to the other apes here in brown. They're not, in terms of their gut microbial community similarity. It's more closely tied to the Cervipithecine monkeys, even though our closest phylogenetic relatives are apes. We have a baboon-like gut microbiome. This is rather surprising. My slides are magically moving ahead without me. This lack of phylosymbiosis may have to do with human dietary diversity and habitat diversity. Both humans and baboons evolved in similar environments. They both have omnivorous diets with high dietary diversity. But I also want to point out that this data set is a little limited, and I'm comfortable pointing that out because it's my work. This data set actually doesn't have that many omnivorous monkeys in it. The omnivores are really just these three species, 2 species of baboons and a species of red-tailed monkey. And it really is missing omnivorous monkeys from outside of Africa. There are no omnivorous platyrrhines. And there are no omnivorous platyrrhines, and there are no omnivorous monkeys from Asia. It really just is baboons and other monkeys from Africa in here. And I'll get back to that in a minute. But this primate data and this fact that the omnivorous primates sort of converge in terms of their gut microbiome mirrors data from another recent paper that looked at the gut microbiome of animals more broadly. And what we see in this paper is there's kind of a weird convergence of bats and birds here in purple and yellow. And these gut microbiomes of these bats and birds, most of the species in this paper are heavily insectivorous. They seem to not be terribly host specific. They don't follow this pattern of phylosymbiosis where microbiome similarity mirrors host phylogeny. Instead, what we see is that there is a lot of inter-individual variation. So each of these dots is an individual's microbiome. And you can see that there's a wider spread of bat and bird microbiomes than there are for any of the other clades of animals that they looked at. And this increased inter-individual variation might mean that there's more plasticity in the microbiome of these insectivorous animals. There are their individuals, even within the same species, have gut microbiomes that are more dissimilar from each other than you would expect based on what we know about the gut microbiome of other animals. And we also see this increase of inter-individual distance in the primate data we have. So this is when we zoom in just to the humans and cercopithecines and apes. What we see is that humans have greater inter-individual differences, greater beta diversity than we see in any of the other primates. And in fact, even if you look at humans in the same population, that you see greater distance between the gut microbiome and any two humans in that population than you see in all of apes and cercopithecines combined, which is a little wild. There's a lot of variation between individuals in the human microbiome. So all of these pieces of information have made me start thinking about a couple of questions. And specifically, so how does omnivory or insect feeding influence the gut microbial community structure of non-human primates? Do we see this same pattern across primates more broadly if we include more omnivorous species, where we see that the gut microbiome converges on an omnivorous gut microbiome that looks different than that of other primates? And does eating a more diverse diet, does having higher dietary diversity influence gut microbial plasticity? Is that what's driving this large inter-individual difference in the gut microbiome? Because the gut microbiome is responding to nutrient intake changes. So if your nutrient intake is constantly changing because you're eating a wide variety of foods, that might cause greater beta diversity in your gut microbiome. And these are questions that have been sort of percolating in the back of my mind for a really long time, actually. They circle back to a conundrum that I encountered during my graduate work on white-faced capuchin foraging ecology. So this is me 10 years ago in grad school studying white-faced capuchins in Costa Rica. You can see it was a long time ago, but so I studied a population of white-faced capuchins in northeastern Costa Rica. So way up here, this is tropical wet forest, it rains year round. And I was interested in how insect feeding is shaping foraging behaviors. White-faced capuchins eat a lot of insects. About half of their diet is insects, so they were a great system in which to study this question of insect foraging in primates. And sort of as a side project, we decided to sequence the gut microbiome of these monkeys. We thought, why not? And this was sort of the state of our knowledge of the gut microbiome of non-human primates when I was in grad school. We didn't know much at that point. We had some data from spider monkeys and black and white colobus and gorillas. We had some howler monkey data in the lab that hadn't been published yet. And what we thought was that non-human primate gut microbiomes are primarily composed of firmicutes here in blue, and Bacteroides in green. This is also what we knew about human gut microbiomes at the time. They were mostly composed of firmicutes and Bacteroides. So this is the primate gut microbiome pattern. And so when I analyzed samples from my dissertation subjects to look at their gut microbiome, this is what I found. And it did not fit the primate pattern. We found this really high relative abundance of proteobacteria. This was really surprising because there wasn't a lot of firmicutes and Bacteroides. And also the literature at the time thought that proteobacteria in the gut of a mammal was probably pathogenic. So we have all of these potential pathogens. I actually showed the data to Andres Gomez, who was provided this gorilla sequencing data, and he looked at my data and he said, "Liz, are your monkeys dead?" I said, no, these are wild monkeys running around the forest, perfectly happy. And he was like, "Liz, they have to be dead. You can't have that much proteobacteria." But that is what the gut microbiome of a capuchin looks like. This was one of the first times we had looked at the gut microbiome of a primate that consumed a lot of insects. And so it maybe shouldn't have been surprising that we found something different, but we were surprised. Which leads me to this question. Is it just white-faced capuchins that are different and have all of this proteobacteria? Or have we just not looked at enough insect-feeding primates at that time? Were we seeing something weird or not? So this project is in collaboration with Katie Amato, Tony Di Fiore, Andres Link, and Eduardo Fernandez Duque. And we have collected samples from a single clade of flat-eyed monkeys. These are monkeys that live in the Americas for this project. We have data from 10 different species of flat-eyeds, most of whom live at the same national park. So we've constrained our geographic range. So we have samples from owl monkeys in Argentina, samples from one species of tamarin in Bolivia, and then samples from 8 different species of monkeys at Tipitini Biodiversity Station in Ecuador. These are the monkeys that we've sampled. So we have these 2 species of tamarins, we have white-fronted capuchins, squirrel monkeys, owl monkeys, red titi monkeys, Kurosaki monkeys, woolly monkeys, white-bellied spider monkeys, and red howler monkeys. And the literature classifies 4 of these monkeys as omnivores. They eat a substantial number of insects. The way we measure this is the percentage of their feeding and foraging time that is spent feeding on insects. So at least 30% of the feeding and foraging time in the literature is spent eating insects. And then the rest of the species are classified as frugivorous or folivorous. However, these are based on the literature and they might not be that accurate in terms of what these animals are actually eating. I'm happy to talk to you about that later. Okay, so with this data set, we're asking 3 basic research questions. Do omnivorous flat-eyed monkeys have a gut microbiome that is different from those of other flat-eyeds? And then do they have a gut microbiome that's more diverse? Do we see higher alpha diversity? Do we see larger inter-individual differences? And then do we see the same enrichment for proteobacteria that we see in white-faced capuchins? So we'll tackle this first question. So just give you a brief run through of what we do with these samples. These samples were collected in the field over a number of years, over the past 12 years, actually. So they represent a wide range of time. Most of the samples were collected in the summer, our summer, so June through August in Ecuador, but not all of them. There are some that fall outside that range. All of the samples are from adult individuals, though. We extract DNA from the samples and then we amplify various things. So for the microbiome data, we're looking at 16S RNA sequences as well as shotgun sequences, and we've done qPCR to look at butyrate gene abundance. Butyrate is a short-chain fatty acid that tells us something about microbial metabolism in the gut. And then we've also done some diet metabarcoding. We've looked at invertebrates, plants, and vertebrates. And then this is our data analysis pipeline that is not that exciting to talk about. Okay. So in this set of samples, is diet or phylogeny more important in structuring microbiome composition? And the answer, unsurprisingly, is both. Both diet and phylogeny structure microbiome composition, and the R-squared values for both are similar. Between 7 and 10% of the variation is explained by diet and or phylogeny. What does pop out as interesting, though, is that these pink individuals are all of our omnivorous primates. And there is a huge spread of diverse inter-individual diversity. Individuals are, again, there are a high number of differences between individuals, especially when compared with the folivores and the frugivorous monkeys. And we see the same thing when we look at gut microbiome composition. This is with the shotgun metagenomic data. These is functional pathway abundance. So all pathways that are present across the gut microbiome that the microbes are able to carry out. And you see a similar thing where diet and phylogeny both impact function, but you see greater individual differences between omnivores compared with frugivores or folivores. And then the gene family presence data shows the same thing. And if we look at our butyrate qPCR data to look at the specific microbial metabolite, we see that omnivores, here again, pink, have much higher butyrate metabolism than folivores or frugivores. And this is compared to the phylogenetic differences, where we do see that CBD, which is where all of our primates in CBD are omnivores. So they have higher butyrate pathway abundance as well, or butyrate production as well. So these omnivorous platypi monkeys do have a distinct gut microbiome. We do see structuring by phylogeny as well, but it seems the diet is playing a really important role. And we see greater inter-individual differences between omnivorous monkeys. But is their gut microbiome more diverse within individuals? One would think that if you're eating a greater diversity of foods, that you would have a really high diversity of gut microbes, but they don't. So the individuals with the most diverse gut microbiome are the folivores, who are consuming a lot of leaf matter and are really heavily dependent on their gut microbes for breaking down cellulose and other plant polysaccharides. And the omnivores have the lowest diversity of microbes in their gut. And again, we see an effect of phylogeny as well. And this, I'm showing you only Shannon diversity here, but this pattern holds for multiple metrics of species richness and diversity, where omnivores have a lower diversity of gut microbes. So omnivorous platypi monkeys don't have a more diverse gut microbiome. Are all omnivorous monkeys enriched for proteobacteria, or is it just white-faced capuchins? Well, there's a lot of variability across these monkeys that we looked at in their gut microbiome composition. There is no one single primate gut microbiome. There's a lot of diversity of which gut microbes are present, but these are our omnivores, where these are our capuchins here. They do have a large amount of proteobacteria, like the capuchins in Costa Rica. So that does seem to be a feature of that genus. These tamarins here have a large amount of proteobacteria, but this other species of tamarin does not. And then our squirrel monkeys here, who are our fourth omnivore, have a lot of proteobacteria, but it's not more than these other two species here, socky monkeys and PD monkeys, that are classified as frugivorous. So it doesn't really seem like proteobacteria is necessarily a feature of all insectivorous or omnivorous primate microbiomes, but is maybe a feature of some of them. There are some really consistent taxonomic differences in which phyla of bacteria are present based on dietary pattern, though. We do see an increase in actinobacteria in omnivorous primates, as well as an increase in Bacteroidetes in these frugivorous primates. That's likely due to the fact that a lot of Bacteroidetes species are involved in sugar metabolism. And then we see that there's less cyanobacteria if you're eating fewer plants, which probably means that our filtering methods for getting rid of chloroplast sequences are not robust enough. And then we see higher proteobacteria in some omnivores and higher canericutes in some folivores. So some omnivorous monkeys have more proteobacteria in their gut, but not all. But I really want to focus on this middle question. This question of if omnivorous platypi monkeys have more diverse gut microbiomes. Why do they have such low within sample diversity? We see this really high inter-individual diversity, or beta diversity, and this relatively low within sample diversity, or alpha diversity. which leads me to this question of does dietary diversity actually vary between folivores, frugivores, and omnivores, or is this just an assumption I made going in? This seems like a really simple question, but we tend to assume that species with more generalist diets have more diverse diets on a day-to-day basis. But just because you can eat more kinds of food doesn't mean you do regularly or that you do on any given day. The other assumption that I've made is that dietary diversity is somehow related to gut microbiome diversity, and it may not be in every case. So this is where we get to that DNA metabarcoding data. And we can look at the diversity of plant, invertebrate, and vertebrate species that are eaten by each of these different dietary categories of primates. We don't see any difference in the diversity of plants individuals are eating based on whether they're an omnivore, frugivore, or folivore. Everyone is eating a lot of different species of plants, whether those plants, they're primarily eating leaves or fruit or some other plant part. They're all eating a lot of different plants. We do see a higher diversity of invertebrates being eaten by omnivores compared to folivores, but we see the same thing for frugivores. They also have a high diversity of invertebrates that they're consuming, at least the way we've classified them in this study. I'm sorry that it keeps doing this. And then finally, we see that, again, omnivores and frugivores are eating a lot more vertebrate animals than folivores are. Monkeys, when they're eating vertebrates, tend to eat lizards, bird eggs, and then some small mammals. And the folivores really are a little bit too slow to catch those fast little creatures. So it seems like omnivores are eating a really high diversity of invertebrates and vertebrates, but not any higher than we see the folivores or the frugivores consuming. And then if we look at associations between plant and vertebrate dietary diversity, we find something a little confusing, in my opinion. So what we see is that there is a correlation, a positive correlation between the diversity of plants you eat and the diversity of microbes in your gut. And there's a lot of mess around that line, but there is a relatively strong correlation there. We don't see any correlation between the diversity of invertebrates you eat and the diversity of microbes in your gut. And we see a negative correlation between the diversity of vertebrates you eat and the microbes in your gut. So dietary diversity is related to gut microbiome diversity, but only for specific portions of the diet. And we actually see an inverse relationship when you're consuming vertebrates. So what it seems like is happening is that, so the diversity of plants and vertebrate prey in the diet are associated with gut microbiome diversity, but in a way that might actually be causing lower dietary diversity in omnivorous species. They are consuming slightly fewer plant species, and they're consuming far more vertebrate species than folivores are. And this might be then driving the lower diversity we see in their gut microbiome. So this gives us sort of a clue as to what's happening here with alpha diversity or within sample diversity. But it's not really explaining this large amount of inter-individual variation we see in the omnivores. It's not explaining our beta diversity results, where we see that two individuals of the same species have wildly different microbial communities. So it's possible, we're pulling samples from different times of the year, that diet composition might be more sensitive to seasonal variation and resource availability in omnivores, which lead to variation in the gut microbiome. So is this high inter-individual variation in omnivores related to seasonal diet variation, where during some times of the year, fruit is more available and some times of the year invertebrates are more available. And as they make these large scale shifts in diet composition, their gut microbiome composition follows. So I'm going to show you a little bit of data from black howler monkeys on the left and white-faced capuchins, and then I'm going to show you a lot of data from common marmosets there in the middle. And this project is in collaboration with Julio Beacon Marquez, Chrissy McKenney, Katie Amato, Paul Garber, Antonio Salto, Nicola Scheel, and Peter Finnegan. Takes a lot of folks to run these big microbiome projects. Okay, so first we'll look at data from black howler monkeys that shows that seasonality is really important in structuring the gut microbiome. We know in these monkeys that there are large seasonal shifts in diet that result in large seasonal shifts in gut microbiome composition, where when individuals are eating a lot of fruit, their diet or their gut microbiome looks a lot different than when they're eating a lot of leaves. And we see this reflected when you look at gut microbial metabolism. So this is metabolome data. Gut metabolites are metabolic products that the gut microbes make when they're doing various metabolic functions. So it's a measure of looking at what actual functions they're doing. And we see that strongly shift across our 3 seasons. And these shifts roughly match the changes in nutrient intake, where when energy and fiber intake increases, we see that the gut microbes are spending more time breaking down glycolipid and fatty acids. And then when non-structural carbohydrate consumption increases, we see more starch and sucrose metabolism. Where it gets a little interesting is for protein intake, the gut microbes are seemingly buffering a decrease in protein intake by upregulating their amino acid metabolism pathways. But we see this really strong seasonal pattern in howler monkeys. But we don't see it in white-faced capuchins. So this is the gut microbiome across periods of high and low fruit consumption and high and low invertebrate consumption. And you can see that their gut microbiome basically looks the same year round. But this is a population of white-faced capuchins who live in an aseasonal forest, so their diet isn't actually shifting that much. High fruit consumption period means that 50 to 60% of their diet is fruit. Low fruit consumption means that 30 to 50% of their diet is fruit. And then a high invertebrate consumption period is about, again, 50% to 60%. And low period of invertebrate consumption is still a lot of invertebrates. And again, this is an aseasonal forest, so we see a lot of rainfall year round. There's always a lot of fruit available. There really aren't large fluctuations in food availability. And so we don't see seasonal differences in white-faced capuchins who are an omnivore, but we don't really know if that's because there's a lack of seasonality where I studied them, or if this is a feature of omnivores where dietary differences aren't influencing seasonal variation in the gut microbiome. So we're going to look in common marmosets, which are an omnivorous monkey that lives in a highly seasonal environment. In this case, see, these are marmosets that are living in a Caatinga environment in northeastern Brazil. Part of the year it looks like this, like a desert, and part of the year it looks like savanna woodland. About a third of their diet is insects. And again, they experience the stark seasonality. So this is where this field site is here in northeastern Brazil at Veracruz Biological Station. These samples were collected a couple of years ago. And we collected fecal samples both during the dry season and the wet season in 2 subsequent years. And we know in this population that there are dietary shifts during the dry season to account for the fact that there isn't much food available. They are consuming more cacti and lizards during the dry season, as well as consuming leaves, which marmosets don't consume in tropical forests normally. And again, similar methods. We looked at microbiome composition and function, as well as doing diet metabar coding. And what we found that even though there's this really strong seasonality in this environment, we aren't seeing seasonal shifts in the gut microbiome. So the season in these graphs is denoted by these 2 ellipses, which you can see are almost entirely overlapping. And in fact, the variable that is significantly influencing the gut microbiome is which social group individuals belong to. That's accounting for 20 to 30% of the variation in the gut microbiome, which is a lot for the gut microbiome. If we look at gut microbiome function, we see a similar pattern where season is not related to gut microbiome function for the most part, but social group is strongly structuring the function of the gut microbiome. We do see some differentially abundant functions across seasons. If we use left-C to look for differential abundance of functions, there are 33 different functions that are either enriched in the wet season or enriched in the dry season. This we see far more between social groups, but there are some pathways that might point to the gut microbiome making slight functional shifts related to diet. During the wet season, when these marmosets are consuming a lot of fruit, we see some pathways related to simple sugar degradation and sugar alcohol degradation and protein biosynthesis being upregulated. Fruit is actually full of a lot of protein compared with other food sources that primates eat. And then during the dry season, we see pathways related to immune system evasion and vitamin K biosynthesis being upregulated. But there are a lot of basic bacterial functions that differ between the two seasons. The other 13 pathways really are just related to things that bacteria do as part of their normal life cycle that probably aren't related to seasonal differences in nutrient consumption. So why are we seeing that social group and not season is really structuring the gut microbiome in this population? One of the reasons is that marmosets have really highly cohesive social groups. They have small social groups, and they spend a lot of time in close contact with each other or in the same plant species, like the same tree. And so it could just be that individuals are passing microbes back and forth really readily through horizontal transfer, or that they're eating very similar diets, or both. It also could be that we don't have a great, that the seasonality of diet and diet microbiome interactions isn't as great as we assumed, where we do see these shifts in the dry season where they're eating some more vertebrate prey and some additional leaves, but the rest of the diet isn't changing as much as we thought. And so we use diet barcoding data to answer this second question. And what we see is that dietary diversity doesn't differ a lot by season. We see this slight increase in the diversity of invertebrates consumed during the wet season, but the diversity of vertebrates and plants remains unchanged. And if we look at the composition, we see something similar. There isn't a large difference. So this is the relative abundance of barcoding sequences that get assigned to either plants, invertebrates, or vertebrates. So it really, you can't compare across categories in terms of the proportion of diet because there are differences in the primary efficiency. But within the dietary categories, you can see that they're consuming about the same amount of vertebrates in each season. Maybe a few more invertebrates, but the difference isn't actually significant. And then plant consumption is equivalent across seasons. We also see that in this population, dietary diversity and microbial diversity are not at all related. The diet doesn't really seem to be structuring the gut microbiome on a day-to-day basis. So we don't see any correlation between the Shannon diversity of the microbes and of the plants consumed or of the invertebrates consumed. And if we look at gut microbiome composition and dietary composition and compare them using a Mantell test, we also don't find a significant correlation. There just really is no relationship. So in these common marmosets, social group but not season is associated with gut microbiome composition and function, which was not what we predicted would happen. And the diet and the gut microbiome are not strongly associated, which again is not what we would predict. So it seems like dietary variability over longer temporal scales like seasons is not really driving the high interindividual variability we see in the gut microbiome of omnivores. Okay, so I told you I had a destination and we are finally getting there. So what we see is that omnivorous primates do have this distinct gut microbiome that not only differs in composition, but has this high inter-individual diversity and relatively low within individual diversity. And then I haven't really shown you strong evidence that gut microbial diversity is related to dietary diversity in omnivores at all, at least not the invertebrate component of their diet. These decrease in within individual diversity is not related to the diversity of invertebrates in the diet, and the increase in inter-individual diversity isn't related to seasonal differences in insect consumption. So what is going on? Why do omnivorous primates have this high inter-individual variation? And sometimes a lot of proteobacteria. And that's linked, I promise. So I would have loved to have shown you a neat little story where I could say that omnivory and eating a high diversity of foods leads to a more diverse and variable gut microbiome. That's not what we see. So why are omnivorous primates weird? I spend a lot of time thinking about this. And I think at this point, and I might be wrong, there might be other options, but I think there are two options. And they're both related to proteobacteria. One is related to what proteobacteria is doing once it's in the gut. Some proteobacteria are involved in lipid metabolism. Insectivorous and omnivorous primates are eating a lot of insects which have high lipid content. So it could be that this increase in proteobacteria is allowing them to break down these lipids and increase energy availability. But that doesn't really explain the high inter-individual differences. The other option might. And there are three sort of disparate pieces of evidence to get to this conclusion. The first is the gut transit time of omnivorous platyrrhines is really fast. It's 2 to 4 hours from when food enters the mouth to when it comes out the other end. We're able to measure this using seed passage rates, and this has also been experimentally validated in labs. So the food isn't spending much time in their gut, which means that the bacteria are probably making it through alive, or at least not totally broken down. The other thing is the spatial distribution of insects, particularly in tropical forests. The spatial distribution of insects as a whole is really heterogeneous. You're going to have a lot of different species of insects scattered across the landscape, and you'll have some areas that have a large amount of one kind of insect and other areas that have a large amount of another kind of insect. And monkeys are opportunistic predators. They will eat any insect they come across, including the ones that sting them and bite them, if it looks like it's going to provide them energy. So there's a high inter-individual variability in which species and even individuals of insects a monkey is consuming. And the third piece of data is the composition of the gut microbiome of insects is really highly variable, and also a lot of insects have a lot of proteobacteria. So this is data from 6 species of grasshoppers at a single geographic location. Gray in these graphs is proteobacteria, so you can see that some of them have a lot of proteobacteria, some of them don't, but that there's a lot of variability in the microbiome consumption of the insects themselves. And even within a species of insect, you have a lot of variability in the microbiome between individuals. So this is a little bit hard to see, but this is data from a bunch of different species of Drosophila. But what I want to point out is that these are 4 different individuals of Drosophila melanogaster, and then 2 of a different Drosophila species, and so on and so forth. So there are multiple individuals from each species here. And you can see, even within melanogaster, there's a lot of inter-individual variability in what microbes are present in their gut. So what bacteria are present in the gut of the monkeys is likely highly dependent on which insects are being consumed. And not just which species of insects are being consumed, but the actual individual insects. Which leads to the question of what is in the gut of the insects that these monkeys are consuming. Well, during my dissertation, I happened to trap insects. So I have a bunch of insects in the freezer that are from this area where I was studying white-faced capuchins. So what we're going to be doing going forward is looking at the microbiome variation of the insects I trapped during my dissertation to see how variable the insect microbiomes are, and then using that data in combination with the gut microbiome data from the white-faced capuchins themselves to see if the insects are actually seeding the gut microbiome of the white-faced capuchins. So this is one of the projects that my lab is going to be working on over the next little bit. So if you're interested in this sort of thing, I am taking grad students. And with that, again, there are a lot of people to acknowledge. These are really big studies that involve a lot of people working with them, both in the field and in the lab. And yeah, I'm happy to take any questions.
Okay, we already have some other questions.
Hi, thank you for your talk. That was really great. I have two questions. My first question has to do with looking at the relationship between dietary diversity and gut microbiome diversity. And oftentimes we look at the microbes as basically making nutrients available to the host that would otherwise be unattainable. But given that these primates have two versions of chitinases, is it possible that they're able to digest them themselves, so the microbes wouldn't actually reflect the dietary diversity wouldn't actually impact the gut microbiome if they're not depending on these microbes anyway to digest them?
Yeah, that certainly is a piece of the puzzle. They are not depending on their microbes to digest chitin. And so we wouldn't have those microbes that are chitin specialists in their gut, but we might expect them to see microbes that are tuned to digesting other nutrients and toxic compounds that are present in the insects. And we haven't done a deep enough dive in order to look at that.
And my second question is, interesting this. So I know that common marmosets are obligate gumivores, so they like to eat these tree wounds, and then usually if you find where a social group is feeding on gum, you can go there every day and that social group will be there eating. And so I was wondering if in Katinga they do that, and if that could be driving variation between social groups, like the actual microbes that are on these trees that they're regularly feeding on, if they are obligate gumivores there.
They are. And we have fungal sequences, ITS sequences from this population that I have an undergrad working on right now.
Would the gum show up in the metabarcoding data from the fecal samples? That would.
It would in the ITS sequences.
Oh, okay.
Yeah.
Cool.
Yeah.
All right, thank you. That's really.
Oh, sorry. Be back there.
Hi, thank you for that great talk. I was curious about your last point you were making, or you were speculating that perhaps the variation in the insects, in the gut microbiomes of the insects themselves could be, you said, seeding the microbiome of the primates. And I was wondering how or if you can tell whether that's actually colonizing the gut microbiome of the primate, or whether that's just passing through and you're sampling that microbiome of the insect.
Oh, yeah. So we can't necessarily tell if they're becoming, if they're colonizing without taking some colon biopsies. We do know that there is a difference in the microbiome that's represented in a stool sample versus what is represented in an internal sample, but they are at least correlated. So it's likely, I don't know, maybe, but maybe not. I think, Rick had something.
Well, given that I'm a reductionist,
Yes.
and that is, can you do the experiment where you actually precisely define their diets, or define the microbes that they're encountering, and see if you can't shift it like this thing with all the proteobacteria? What happened to the fed them a diet where they almost had none?
Yeah.
As opposed to lots.
Yeah.
And then look to see how it varies amongst individuals as well as the whole population.
Yeah, we could certainly do that in captivity. Monkeys are generally, even omnivorous monkeys are generally not fed insects in captivity, but you can introduce insects into their diet to see if that is shifting their gut microbiome. And then there's actually somebody at Nebraska who is creating a germ-free marmoset model where he's going to be able to artificially introduce microbes and see what happens.
It just seems like these field experiments seem to be very messy.
They are messy.
And so if you want to sort of, tip the scales in your
Yeah.
favor one way or the other.
Do we have any other questions?
Thank you. Can you hear me?
Yes.
I was just wondering, in the beginning of your presentation, you were talking about looking at the difference between microbiome diversity in relation to diet and phylogeny. But you also mentioned that was it CBIDs were highly, there was a lot of CBID species that were omnivorous, and so I'm wondering if this group contains is one of your taxonomic categories, but also contains a lot of the omnivores. How did you distinguish, make sure that you're not like confounding phylogeny and diet and analyzing them separately?
Yeah, so we do have a non-CBID omnivore in that sample set. But there aren't any CBIDs that aren't omnivores the way we have traditionally classified them. So that it does make it difficult to pull apart. We actually designed the study to look at encephalization and diet interactions. So we were actually looking for encephalized omnivores and non-encephalized omnivores, which do exist within CBID, which is why we set it up that way. But yeah,
Thank you.
you're welcome.
Hi.
Hi.
So in terms of like putting the puzzle pieces of the microbial composition, you talked about maybe these bacteria could be helping metabolize into lipids.
Mhm.
And I was thinking from other perspective that maybe what if these patterns of bacteria could be influenced by biomic composition. So I was thinking stages, and I was thinking some of these other part of the microbial composition that creates the gut microbes could have influence in how these numbers change.
Yes, they definitely could. And that's actually something, the other time we see a lot of proteobacteria in the gut in humans is during pregnancy and lactation. So this is, Stan's question. So yeah, this is something that I'm going to be looking at in humans with a collaborator where we're going to be able to take samples and store them well enough that we can look at viral composition and look at environment bacteria interactions during pregnancy and lactation.
Yeah, so I'm wondering if you have any sort of environmental samples, because the microbiome doesn't only come from vertical transfer and it doesn't only come from diet. Someone mentioned they like chew tree bark or something, or if they're eating leaves, like there's if an environment is unique and exists on this planet, it pretty much is guaranteed to have a unique microbiome. So I'm wondering if you've got sequences from those as well as diet.
We don't for most of these samples. We do for the owl monkeys though. Yeah.
Thank you. It's a really interesting talk. I had a question about your grant analysis of concepts, which I think is really, really cool. But I was wondering the type of resolution you'd be able to get, since you were talking about how you think the type of insect, also even just individual insects, is so important in your hypothesis in terms of, do you have like resolution in terms of knowing, being able to match what the monkeys that you have samples on were eating at that time with the insect data that you have?
Yeah, so what I will be able to do is look at multiple individuals within an insect species and look at gut microbiome variability there. The DNA metabarcoding data doesn't get me resolution on insect taxonomy fine enough to figure out which species of insects the monkeys were eating. What we will be able to do, though, is a source tracking analysis where we can say that the insect microbiomes are the source. It asks if they are the source for the sink microbiome in monkeys.
Other question?
Oh.
Is it how sort of homogeneous are the monkey poops?
Yeah.
I'm wondering, Do they just, if they eat the insects whole or with minimal chewing, then perhaps some of this might just be from the methods of processing of,
Yeah.
if you blend up the.
Yeah, so I can tell you far more detail about this than you want to know, but no, it's fine. So when monkeys are eating insects, they usually pull off the wings and legs. And when they don't, the wings and legs often make it through whole. But the body of the insect, which is primarily what they eat, does break down. When we look at monkey poo, even for the omnivorous monkeys, unless they have eaten small mammal prey, their fecal samples look like digested fruit because the transit time is so rapid. When they have eaten a mouse or something, it's a whole other story.
Thank you.
So that leads into my question. What is it like to be gathering these samples? Are you out there? Looking, tracking the monkeys, and then as soon as they go poop, you like run under them? Are you tracking them with a crop? Basically behind their butt and to get them, or just coming across the samples?
So we are actually following them because we're collecting behavioral data at the same time. These are individually recognizable monkeys. So we are doing focal follows of an individual. And yes, when your focal poops, you run over and you get it. Yeah.
And then on a day-to-day basis, how much is, I know you talked about seasonality, but day-to-day basis, is that gut microbiome changing across an individual?
It is, and I haven't done a really robust longitudinal analysis to be able to quantify it, but it is pretty variable. Yeah.
So if I'm eating a mouse, it's going to change versus feeding on insects or fruit that day.
Yeah.
Have you noticed, like, does the microbiome, is it different between like social groups at all? Like,
So in the marmosets, it is. That is the only population where I have more than 2 social groups. But other folks have looked at this in other species of primates, and they do find social group differences.
Hi, I have a basic question.
Yeah.
I was wondering why you're just looking at the gut microbiome of the insects versus microbiomes throughout the entire insect.
Oh, it probably will be throughout the entire insect. Yeah.
Oh.
Sorry.
Hi, so I am no expert in primates or their interactions, but I was wondering if it's possible that the similarity in gut microbiome composition among the marmoset social groups could be related to grooming behaviors or something?
It could, because marmosets are highly social individuals, unlike, say, a howler monkey, which just sort of sit near each other. Yeah. So yeah, social behavior is a big component of what is, how microbiomes are getting structured.
I guess like consuming the insects present on. farmers, their
yeah.
social
Yeah, the parasitic insects. Yeah. I hadn't thought of that, but yeah, that would be really interesting.
Okay, we've gone over our time, so let's thank Dr. Mallot. And please join us across the hall for reception.